Hey everyone, it's Mr. Sinti, and today I'd like to talk to you about mutations. And in particular, I want to talk to you about point mutations. Point mutations are different from other kinds of mutations because point mutation is when just a single base pair is altered in the genetic material, and that's as opposed to some large-scale mutations. Maybe this will come up in a future conversation. Things like if a whole segment of DNA is translocated and excised from one area of DNA, one chromosome, and travels to another one like a jumping gene, or if portions of the large portions or sequences are duplicated, and even some can be inverted and switched around. You can imagine those will have some drastic effects on the phenotype. But today's point mutations, and we're going to talk about how point mutations can affect protein because that's what it's really all about. And so let's jump right into this if I can. And so let's see if I can make the screen a little bit larger. And so mutations are changes in DNA, and so you can simply look at that, a change in DNA. Now, mutation has a negative connotation, meaning that mutation, it's mutated, meaning like something's wrong with it, but it simply means that it's a change. And a change could be no effect, there could be a positive effect, and it could be a harmful effect. So these are the three possibilities we're working with when you change the DNA sequence, the nitrogen-based sequence. The truth is, the only way that you could ever improve, if you will, the only way that you're going to make an improvement onto a protein in terms of protein structure and protein function is you've got to make a change to your recipe, which is ultimately going to result in a change in RNA, and then that's going to result in a change in the protein. It's going to change the primary structure, which is the amino acid sequence, and as a consequence of that, the whole molecule will be affected, and therefore its function. So let's take a look at that. I think it's certainly worthy. And obviously, mutation is the raw material for variation, which is rather critical in biology when you consider the evolutionary implications of variation. So, as I mentioned, these are going to be point mutations, or a single base pair. So, one of the first things that I want to talk about and emphasize is the fact that when a mutation occurs to a gamete, which is a special cell, in the male it's a sperm, in the female it's an egg, when there's a mutation to those cells, that has implications to future generations, meaning that those changes that occur when the egg is fertilized to become the zygote, that means the zygote will ultimately become the embryo, the fetus, and the baby. So that's going to carry on to the next generation. It's more serious by far if a mutation were to occur to, just say, anything on your body, like your skin, like a somatic cell or body cell. Certainly there's implication in terms of you, the organism, but that's not inheritable. So that's really important. So when we talk about changes in a mutation resulting in a change in a phenotype to the offspring, we're talking about gametes. So that's important. So let's see. Let's give one of the classic examples of all. This is like the example found in most textbooks in biology about mutation. And it happens to be with sickle cell disease. Sickle cell is referring to the shape of the cell like a sickle, and it has to do with a red blood cell. Let me change this to red. So a red blood cell, a red blood cell is normally sort of like this, and I have a little video coming up in a moment, but it's kind of a biconcave disc, and a sickle shape has this appearance, like that, sort of like a crescent moon. And the consequences of that are profound. It's a good example of one little mutation leading to many different phenotypic consequences, something known as pleiotropy. When we get into inheritance a little bit more, we'll talk about that. So let's consider this. Here's the wild type hemoglobin, meaning that the gene for hemoglobin is really long. So it goes on to the left very far and goes on to the right very far. But this is just one codon right here. Wild type is sort of the normal. As you may know, C bonds with G and A. So this is the normal wild type codon in messenger RNA. And then you can see down below the normal amino acid for in this one little area in the protein hemoglobin. Now, hemoglobin is the protein inside red blood cells, and there's about 300 million molecules of this. This is the protein that carries oxygen, so it's rather critical. And so this amino acid, glutamic acid, helps to shape the hemoglobin just the right way to hold oxygen. But if there's a mutation, do you see here? This is what we mean by a mutation, a change in the sequence. Instead of it being CTT, it's CAT. So there's the change. There's been a base change. And if the base is changed to an A, that means the messenger RNA is going to be changed to a U. So the codon's no longer GAA, it's GUA. And as a consequence of that, when you look at your decoder, the amino acid that it's coding for is different. Instead of it being glutamic acid, it's valine. And you might be thinking, no big deal. One amino acid, no one would ever notice it. Well, maybe it is no big deal. Maybe the amino acid is similar in terms of polar, nonpolar, and it doesn't have much of an effect. Or maybe it's in a region of the protein that isn't as important as another area, for example, the active site. But in this case, it is. It has a different property to it than glutamic acid, and there's dramatic consequences. And so let me jump to that and show you a little bit of what I mean by this. Let me jump back to this and come over here if I may. This is a blood vessel right here, and inside blood vessels is some tissue on the side. This is where your red blood cells are flowing. So when you click on this, if you were to be able to peer inside your capillary, right in here, or blood vessel, you'd be able to see the red blood cells. This is the plasma over here. This is a red blood cell. Inside here, there's the protein hemoglobin. And when it's misshapen, it makes the cell look sickle. And you're like, well, what's a big deal with that? Sometimes it blocks up small blood vessels like capillaries, and then oxygen isn't going to be able to travel to that tissue behind it, and it can cause a lot of pain as a result of that. It's all because in the sequence, what was supposed to be one nitrogen base turns into another, and as a consequence of that, that means that valine is put in instead of glutamic acid, and instead of the hemoglobin being normally shaped, it tends to form these sort of polymer-like structures which alters the entire shape of the cell. And let me back that up just for 1 second. Not only does it affect the ability for the red blood cell to travel in the circulatory system, but I happen to know that the spleen tends to consider these cells like beat up and worn down. So the spleen, inside the spleen, there's white blood cells that attack these cells and remove them from the circulation. So you end up being lacking red blood cells. And so that's why you always hear that association, sickle cell anemia, which is a lack of red blood cells. I'm going to just point that out. And so if we go back over here to the class notes, I want to show you this for a second, just to refresh you. We're talking about a change in the, well, if I can make that a little larger, we're talking about a change in the DNA, but as a consequence, messenger RNA is ultimately the one that is altered. And ultimately, RNA is the one going out into the cytoplasm to make the protein. So this is what we're talking about. We're talking about how messenger RNA is also mutated. And then when messenger RNA goes to the ribosome is just going to make whatever the messenger RNA codon tells it to do. And so this polypeptide is going to have different amino acids. That's going to affect its shape. That's going to affect its function. Maybe even something weird could happen, like for example, a premature stop codon could appear, and therefore the protein is a little bit shorter, and therefore it'll be non-functional. And so this is what we're talking about. It's a change in DNA, but it's really a change in RNA, which is really ultimately, when we're talking about the phenotype, we're talking about a change in the polypeptide structure therefore the polypeptide function. And so here's a close-up of hemoglobin. I really like this. It shows the quaternary structure. There's more than one polypeptide chain here. And then inside this protein are these 4 heme structures. Here's a close-up of the heme, and inside the heme is this one iron atom, and that's what binds the oxygen. So that's why it's important to have iron in your diet, just as a little side note there. And so here's a cool scanning electron micrograph. These are normal red blood cells, and this one is sickled because it has the wrong amino acid. So that's rather serious, a mutation. So you know, when on the surface, you might be thinking, sickle cell anemia, this is an example of a harmful mutation, but you can't jump to conclusions on this because in a future video or in class, I'm going to tell you that there's an example of how sickle cell anemia could be an advantageous phenotype in certain environments. Like, I can't see how that can be. Well, I'll just give you a little forewarning. It has to do with malaria, and being bit by a mosquito carrying malaria, it can be lethal, but if you have sickle cell anemia, it's not. And so that's something, a little forewarning there. So at this point, I want to talk to you about the different kinds of mutations, and the one I've been sort of emphasizing, an example of sickle cell is something called a base pair substitution. And again, it's a point mutation, and a base pair substitution means just that. Instead of one base, another base is substituted for it. And so what are the implications of it? It could be silent, meaning that if the change happens to be the third base, for example, of a codon, it might very well code for the same amino acid because of the redundancy of the genetic code, if you remember that. Like, for example, if you had the codon, I happen to remember this, CCC, it's proline, but if it's CCU, it's also proline. So a mutation may occur, but it's like silent. It's not noticeable. And then sometimes, this is good fortune, I guess. If you switch the amino acids and it has the same property, that's pretty good. And then sometimes a mutation will occur in an area, this is getting a little bit more dangerous, where it's not as important. You might be able to get away with it. There's still a mutation, but it's silent. Now, the other possibilities are that if a incorrect amino acid is put in, that could be detrimental, that could be deleterious or harmful, like, for example, in sickle cell anemia. And so this is what we call a missense mutation, is that a different amino acid is put in there, and that can be often harmful. And then something called nonsense mutation is, again, instead of the wrong amino acid being put in there, a stop codon occurs. There's 3 stop codons. If that happens, it's going to be very harmful. There's no good that can come from that because clearly the protein's going to be shorter, and there's really no chance for it to function, especially if it's teeny tiny. And so let's look at some examples of this if we can. Let me come over here and show you this. There's a little animation here that I want to show you about base substitution. It's really good. First thing I want to say about this is that what we're talking about is DNA here. We're talking about one nitrogen base bonding with another one. So usually a purine bonds with a pyrimidine, but here's the deal. Sometimes, like, this is the normal where you have adenine bonding with thymine and there's 2 hydrogen bonds. Every once in a while, something weird can happen. Notice that hydrogen right there. Every once in a while, and this is just can occur, the hydrogen can kind of jump ship from this amino area and jump over to this area of the adenine, and when it does, it's referred to as a telomeric shift. And as a result of that, odd hydrogen bonds can form, and so adenine wouldn't always bind with thymine. It could sometimes bind with cytosine. Let me let that play. And so you can see how it shifts over like that. And so as a result, what can come from this is, instead of adenine binding, what it's supposed to do is A with T, A can bind with cytosine. And similarly, thymine can have a little telomeric shift and it can bind abnormally with guanine. And so here you can see how when there's a mutation, it's passed on to the next generation, because when DNA copies itself, that mutation that will result perpetuates. And so when the DNA replicates, here's the wild type. Let me just pause this for a second. TTC, so that here's the normal codon AAG, and then here's the normal amino acid once it's translated, lysine. Here's the mutant arginine. So as you can see, when the molecule opened up previously, these will be fine, but then this cell right here will have the mutant. And eventually this will catch up to the organism. So a base pair substitution can result in problems. And so I wanted to show that. And so let me show you this while we're here, and I'll come back to the class notes and look at this more in detail. This is another type of mutation. It's still a point mutation, but instead of it being a base substitution, we call this an addition or deletion, or sometimes it's referred to as insertion deletion. And that means that we're going to add a nitrogen base pair or delete a nitrogen base pair. Now that has profound effects, because if you think about it, like if there's one substitution, one base substitution, it's only going to be one amino acid. So here's the normal wild type. You saw that. So here's the DNA right here, and here is the mutation. Let me pause it. Notice here's the normal or wild type. Here we've inserted a cytosine. You're like, shouldn't be a big deal. You mean the gene's very long, thousands of nitrogen bases. Who would notice an additional C? Well, as it turns out, if you insert a base like that, what's going to happen is when the molecule is then transcribed into messenger RNA, which is what you're seeing here, it's going to consider that, and then the implication is in translation, which is where it matters, you're going to see this is what it's supposed to be. Arg, and I think that's threonine, valine, and lysine. What's going to happen here is when you insert something, the codon is going to be altered. And so this results in something called a frame shift mutation. I'm sure if you can see this. But from every codon downstream from an insertion, the amino acid would be different. And so that results in a massive missense mutation. And so it's a frame shift mutation. And so insertion is really harmful. Not a lot of good can come of that, especially because it's random. This is not by design. And so an insertion, again, is kind of obvious, where you add something. And then the opposite of that is just as bad, is when you have something called a deletion. And both of these often form non-functioning proteins. So again, here's your wild-type DNA. And then here's the mutation. Ooh, a deletion. So now, as a result of that, when you remove the base pair, instead of it being the normal nitrogen base sequence and the normal amino acid sequence, again, these two are normal, but every single amino acid downstream from that deletion is altered. So this is a really harmful type of mutation right there. And so let's go back to the class notes on this. And so if I can, click that, make this a little bit larger, go here, maybe. So here's the example of the base substitution. And again, a base substitution is, hey, look at that, we substituted the C in the third base position with a U, and as a result, no effect because of the redundancy in the genetic code. And then, hey, look at this. Here's a base substitution, and we switched the first base of this codon, and it's a missense. It's the wrong amino acid. Could be good, could be bad, not sure. And then sometimes a base substitution can result in a stop codon. That's not good. Look at the tiny little protein. So that would be nonsense. So as I was mentioning just a moment ago, insertion and deletion are really disastrous because it throws off the reading frame of translation. So it's a frame shift mutation, It results in extensive missense, which means every amino acid is incorrect, and probably a premature termination will result. And so here we are deleting something, here we are adding something, and so it's going to throw it all off. It's going to throw the misshape off. Here's something of note. What would be worse if you deleted one base, or if you inserted one, or if you deleted 3? You're like, wow. On the face value, you might think, well, deleting 3 would be really harmful. But the truth is, if you delete 3 of these, you would only remove one of the codons, and only one amino acid would be incorrect. All the others would be fine thereafter. So not as bad as it turns out. And so here's back to our example of hemoglobin. Again, this is a picture from the textbook switching. It's a base pair substitution. You can see it here. Here's the mutant messenger RNA, and there's the incorrect valine. And so what happens is the wild type gets the wrong amino acid. I'm sorry, the wild type is correct. And this is just showing the silent mutation can have the same amino acid because of the redundancy. This shows missense, the wrong amino acid, and the hemoglobin was an example of missense. And then nonsense is a premature stop codon. And then here's another example of the frame shift where you add something or you remove something. So it's insertion deletion. And so let me come back over here for a second, and let me show you this. So ultimately, what I want to sort of drive this point home is the fact that what we're talking about is a change in DNA, which is in the nucleus, but it results in a messenger RNA difference. So you can see here in translation, here's the ribosome attaching. Transfer RNA doesn't know. It's coming in, it's bringing the amino acids, the proteins are forming, the peptide bond is forming here, you can see. And ultimately, where all this is coming down is when the anticodon is trying to bond right over here to the messenger RNA codon. If we can get a close-up right now, we can see here's the correct codon sequence. We're reading here from 5' direction. And so if there's a mutation, you're going to get the wrong amino acid to come in. And the wrong amino acid is going to come in, and what that's going to do is create let me just pause it right there. It's going to cause a change in the protein. And I tried to say at the very beginning of this presentation that sometimes a change is a good thing. So if you change the shape of the protein, maybe, say this is an enzyme, maybe it's working even better than it was before. And so this is how new traits come into place. And it's like, well, wow, it's pretty good. Well, yes. Well, what causes mutations to occur? Well, there's a number of things. One of the things I mentioned before is those sort of odd sort of telomeric shifts that can occur between the nitrogenous bases. But simply DNA polymerase, not to point fingers, but that enzyme can make mistakes. But usually we don't think of it so much because it's also capable of proofreading. It can go back and it can fix its errors. And there's DNA repair enzymes involved, but sometimes mutations get through, and when they get through, they're sort of spontaneous. You never know when they're going to happen. And so, what are the causes of this mutations? Well, I mentioned a second ago that during replication and telomeres, but there's other things. Sometimes there's chemical and physical agents at play, and they're called mutagens, or mutation causing elements or agents. Chemicals, well, let's just go right to the chase. One of the most harmful chemicals is cigarette smoke and smoke in general. These molecules in cigarette smoke can come and strike your cells, especially your sensitive lung tissue, and cause danger and cause mutations to occur. X-rays, so you don't want to expose yourself to a lot of X-rays, and then ultimately ultraviolet light from the sun can cause mutations to occur. And then even some of the ordinary chemistry in the cell, you don't have to worry too much about it, but sometimes when molecules are broken down, free radicals are formed, and those free radicals can cause damage to the DNA. And that's why people like to say that eating fruits and vegetables contain a lot of antioxidants, which they do, and those prevent free radical formation to reduce mutations and ultimately reduce cancer. And so let me just sort of conclude with that conversation here if I can. And this time I'm going to let the video speak for itself. I think I can't really do much better than the narration on this particular video. And so I'm just going to sort of watch it with you if you don't mind.
DNA is under constant attack from reactive chemicals and natural background radiation. Free radicals are the byproducts of normal metabolism in human cells. Seen here as bright particles, they sometimes react with DNA and cause chemical changes.
Ouch.
Radiation can also affect DNA. For example, ultraviolet light from the sun can cause harmful chemical changes in the DNA of skin.
Sunblock is needed.
These changes can lead to kinks in the DNA that prevent genes from being correctly read, or deletions that alter the type of protein produced. Thanks to constant biochemical repair work, most mutations are corrected before they have any effect. But in rare cases, mutations can accumulate, and this can give rise to diseases such as cancer.
But it can also give rise to positive change as well, positive polypeptides and positive phenotypes. So mutation is the source of genetic variation. Thanks for watching.